Welcome everyone, and thank you for attending tonight's Research Tuesdays event, Redefining Human Genetics. I'd like to start by acknowledging the Kaurna people, the original custodians of the Adelaide Plains, and the land on which the University of Adelaide's campuses at North Terrace, Waite and Roseworthy are built. My name is Olivia Bellis, and I'm a PhD candidate in the School of Biomedicine here at the University of Adelaide. Tonight, we're here to learn about the impacts, both positive and negative, that gene modification could have on our health, environment, and society. Imagine a world where advanced gene editing was commonplace. Would there be designer babies, fewer genetic diseases, longer lifespans? While this may sound like a work of science fiction, CRISPR technology is rapidly advancing researchers towards these possibilities. One of the researchers exploring CRISPR's opportunities and possible risks is Professor Paul Thomas. Paul leads the genome editing program and the SA Genome Editing Facility at the University of Adelaide and SAMRI. He has over 30 years of experience in molecular genetics and has published more than 130 scientific articles and more than 12,000 citations. Please join me in welcoming Professor Thomas to the stage.
Great, thank you very much for the introduction, the kind introduction, and it really is a great pleasure to be here giving a Research Tuesday. I've always kept an eye on these seminars, and I've been a member of the university for some time, and I'm very, pleased my number has come up, because it gives me an opportunity to tell you about some really, interesting technology and how it's really changing the way we look at the world in many aspects: health, agriculture, and many other ways as well, which I'll tell you about during the course of the lecture. So a little bit about me, just to put this in context. I'm a University of Adelaide graduate. I finished my PhD here in 1994, so quite some time ago in the Darling building, about 100 metres over in that direction in the biochemistry department. I then spent some time working in London and then some time working in Melbourne, and then rejoined the university back in 2006 to start a research group here. Currently, I'm located at the SAMRI Institute down the road, South Australian Health and Medical Research Institute, commonly known as the Cheese Grater, and lead the genome editing program and the genome editing facility at SAMRI, and also, of course, a member of the University of Adelaide. So I'd consider myself a molecular biologist and genetics researcher. So I've always been interested in the way that DNA works, how the genome works, and how human disease is caused by changes in the genome. And I've always had my eye out for technologies that we could use to restore broken genes, essentially, to make them better and therefore cure people that have genetic diseases. And that's a large part of what I'll be telling you about today. So to put all of this, really in the context of the last 30, 40 years or so and how things have changed. So you can consider the BC era as the before CRISPR era. So CRISPR arrived on the scene back in 2012 as a new technology, and I'll spend a lot of time telling you about how it works. But suffice it to say that before CRISPR arrived, we had very limited options in terms of manipulating the DNA in a cell. So it was difficult to find out what genes did. We could discover genes, it was quite slow, but finding out what they did by changing them was a very difficult, laborious, expensive, time-consuming process. And certainly there was no such thing as gene editing therapy. This was really science fiction. And sometimes I sort of, joke with the PhD students that you could complete my entire PhD on a wet Sunday afternoon with a computer in a couple of hours. And actually, that's pretty close to the truth. So in 2012, CRISPR arrived, and this now brings us to the AD era, and I've called it AD after Jennifer Doudna. Now, Jennifer Doudna, shown here, was one of the pioneers that really got CRISPR going as a technology, and she won the Nobel Prize with Emmanuel Charpentier back in 2020 for the, the application of CRISPR technology. So what we have now with CRISPR is the ability to introduce very precise edits into virtually any cell that you can think of. And this has enabled all sorts of applications that I'll be telling you about today. And these include precision medicines, where we can fix broken genes to make them better as a cure for genetic disease. And it's also greatly enabled discovery science, where we can just find out what genes do by making changes in them and looking at the product of those changes, how it changes a cell's function. So enormous applications. And what's really remarkable, I think, is that from just this initial paper showing that in 2012 that CRISPR could be used to modify cells, within this time period, in late 2023, the first CRISPR therapy was approved by the FDA. So just in this short period of time, we've already got our first therapy on the market. Amazing progress. So what do I want to tell you about today? So here's an overview of what you're going to be hearing about. So first of all, I'm going to have to give you a little bit of a primer on genetics and how genes work and how DNA works. This is just to get everyone on the same page. Then I'll launch into how CRISPR technology works, where it came from, what we can do with it, why we want to use it, and how we're using it. And then, because no talk is complete without a little bit of controversy, I need to tell you a little bit about how CRISPR has changed the way that we can approach all sorts of topics, including manipulating even human embryos. There's nothing particularly special about a human embryo in the terms of its DNA. It's got DNA like every other organism, which means that it can be edited using CRISPR. So there's some very special ethical considerations that we need to think about if we go down that path. And I'll tell you about some of the potential applications for CRISPR and some causes for concern as well. And then lastly, if there's time, I'll tell you about, switch gears completely and tell you about an application of CRISPR that really tackles a wicked problem and that's the problem of invasive species. So invasive species are a real scourge on the planet, and these are two examples. One example here is mice, which is the one that we're particularly interested in. But also in terms of malarial control, there are techniques that we can use that employ CRISPR to control these species and also to control their ability to carry disease vectors. So I'll tell you about that if there's time at the end. Okay, so just as I said, to get everyone on the same page, I'm sure you're a very well-informed audience, but you may not have done Genetics 101. So this is just to give you some background. So the human body is an amazing machine. It contains around 30 trillion cells. And these cells are, of course, the cells that make up every organ within the body, and they have special functions within that organ. And each cell has a nucleus, which is shown here, and what's called the cytoplasm. But the nucleus is the part that is important for this talk because it contains the DNA. So the DNA is made up of individual building blocks. There are 4 building blocks that make up DNA, and these together make up what's called the genome. And it's like a long string of bases all arranged together. And in fact, there are an amazing 3 billion DNA bases, building blocks, that make up the entire genome. So it's a huge amount of information. This DNA or genome is arranged into, it's compartmentalized into sort of smaller and smaller units. There are the chromosomes, and you can think about the chromosomes as being chapters of this book of life. So what I should have said is that the genome is really the instruction manual for the cell. It's the book which tells the cell how to perform its function. So you can think of chromosomes as chapters. The individual genes that we'll be spending a lot of time thinking about today are like sentences that provide instructions to the cell. And these building blocks or nucleotides, A, C, G, and T, are the letters. They're the very code itself that tells the cell what to do when it's interpreted as a gene. And we know from lots and lots of studies that the human genome contains about 20,000 instructions, 20,000 genes to perform its function. So here it is at the very nuclear level. You can see these are the bases or the building blocks shown here in the famous double helix. And a gene is a region along this double helix which contains some information, a sentence that provides an instruction to the cell. Okay, so DNA is very important, obviously, but it doesn't actually do the work within the cell. It doesn't perform specific functions that we know that the cell can perform. For example, it doesn't bind oxygen or it doesn't sense light within the cell. Those functions are carried out by proteins. And what happens within the cell is that the instructions to make those proteins are encoded by the DNA. They're converted to what's called messenger RNA, which is like a transient signal. It's produced from the DNA. It's like a copy of it. Think of RNA as being like a cousin of DNA. And that is then exported out into the cytoplasm where it's translated or interpreted by the cell to make a specific protein with a specific function for that cell. It's important to realise that only some of the genes with any given cell are converted into mRNA. So what I mean by that is that there are 20,000 instruction sets, 20,000 sentences within that instruction manual. Only some of them are used by each cell because only some of them are important for each cell. A blood cell will need to produce hemoglobin to carry oxygen. An eye cell or retinal cell will need to produce the protein that can sense the light. So only some of the genes are converted to RNA and turned into protein. Okay, so we're almost there with the background information. This last piece of information is that we don't just have one copy of this entire genome, we in fact have two copies. So 2 times 3 billion base pairs. And of course we've got two copies, and shown here as chromosomes. We've got two copies because we inherit one from our mother and one from our father. So it turns out that these two copies are not identical. In fact, there's a whole lot of natural variation in the DNA. So I told you that there are 3 billion base pairs. Well, every 1,000 base pairs or so, there's just natural variation. So 999 are the same, and then there's one that's going to be different. So what I mean by that is if you look at the person next to you, you are 99.9% identical at the DNA level to that person, but it's the 0.1% that makes you an individual. It's what makes you different from the other people within this lecture theatre. And so that's the important part. Okay, so what we also know, we know a lot about the natural variation within cell because, in fact, 500,000 healthy individuals have had their whole genome sequenced. And that sounds like a huge undertaking, and it is, because each genome these days costs about $1,000 to produce the entire 3 billion base pairs in their order. So it's an amazing technological feat to be able to do this, but it means that we know a lot about the healthy genome. But what we also know is that sometimes there are rare DNA changes that disable the proteins and cause disease. So what I mean by that is that they'll be located within a gene, so an important instruction set, and they'll change it in some way that the instructions don't make sense anymore. And I can illustrate that in this way. So we're not going to get into the nitty-gritty of exactly how the code works, but you can think of these as two different mechanisms to the way that the DNA is changed and how that impacts the cell and the instructions in the cell. Okay, so what do I mean by that? So changing or deleting just a single base pair or a single building block can have a huge impact on the particular cell. There are changes called missense mutations, and what I mean by that is this where we've got a swap of a letter that makes up this instruction gene. So with this word here, if it's supposed to be how to make a cone cell, so the cells in the eye need to make cones and rod cells, so how to make a cone cell would be a gene within the eye. If this C was changed to a B, it would become how to make a bone cell, which of course in the eye is entirely inappropriate, which means that the eye wouldn't work properly anymore. So that's an example of a missense change, and that can cause disease. A second change, which is a little bit more complicated, but you can think of it as a frame shift or nonsense change. This is where we take away one of the important letters in that sentence. And when that happens, for example, if the sentence was how to make a muscle really, really strong, if we take away this first R, the cell can't read it properly anymore because it becomes how to make a muscle, and then it's essentially garbled after that. And this disables the protein, which means it can't make the protein, disables the gene, can't make the protein anymore, and causes disease. So we know that these kinds of changes are present in at least 6,000 human genes so far, and that number grows basically every day. And together, it's a very common problem, and it causes an issue in around 1 in 20 babies that are born. So now that you're all genetics experts, we're going to talk about how we can develop therapies to address these kinds of changes and how we can potentially fix up these broken genes to make people better again. And that's where CRISPR comes along. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Now, it's quite a mouthful, so I'm not going to say CRISPR again. I'm not going to say the whole lot again. I'm just going to refer to it as CRISPR for the rest of the lecture. And it's made up of a protein and an RNA molecule, which I'll spend a bit of time telling you how it works. But you can think of CRISPR as a pair of molecular scissors. So these scissors can locate a particular region in DNA and generate a double-stranded break. So break that DNA strand. And that's a really useful property, which I'll tell you about how we can use it for disease restoration. What makes CRISPR so really great to use as a lab tool, but as well as a therapeutic, is that it's programmable, which means we can really easily change which part of the DNA it will target. It's very easy to design these experiments in the lab and test lots of different theories about cutting different parts of a gene. And what turns out to be one of the most important properties, CRISPR is really efficient, which means that we can actually get it into human cells within the body and make changes in DNA that will help to restore broken genes. So where did CRISPR come from? So this is quite a long story, and this was essentially the background to the Nobel Prize that was won, in, as I said, in 2020. And like a lot of the tools in molecular biology and genetics research, it comes from bacteria. So the bacterial world has the same problem that we humans have in many ways, in that they're under attack from viruses. So in the bacterial world, the viruses are called bacteriophage, but essentially they're the same as viruses. And these bacteriophage like to inject their DNA into bacterial cells and take over that cell, use the cell machinery to replicate themselves, and then break the cell and go and infect other cells. So the bacteria have evolved defense systems to stop this happening. And CRISPR was really a mystery for a long time until people kind of worked out how this defense system worked. And to cut a long story short, the bacteria have evolved a CRISPR array. It's a whole bunch of genes actually, they're all strung together at a part in the bacterial genome. And it incorporates, sometimes it's able to incorporate a little bit of the DNA from this invading bacteriophage before it gets going. And once it's incorporated into this so-called CRISPR array, that gives the bacteria a chance to defend itself against the virus. And it does this by creating its own little CRISPR molecule that's got this recognition sequence for the original virus, such that if the virus comes again and reinfects the cell, it can target that particular sequence by base pairing, and I'll show you this in a minute, and it's able to essentially chop up that invader so it can get rid of the virus because it knows the actual sequence for it and can cut it specifically. And people realised once they sort of worked out this system that in fact it's not just a single virus it's immune to, but it can incorporate many different viral DNA segments using this CRISPR array. So you can think of it like an antibody. There are many different antibodies that you've got in your body against invaders or pathogens. This is the same thing going on in bacteria at the very level of nucleic acid, at the DNA RNA level. So really interesting bacterial defense system. But what's important for this talk really is that this recognition of a target sequence and generating a break to get rid of that is what's been employed from bacteria, and now we can use this in mammalian cells, so human cells. And that's essentially why these two individuals, Jennifer Doudna, Emmanuel Charpentier, these two great scientists got the Nobel Prize for really applying this bacterial technology and showing that it can work in mammalian cells. So let's talk a little bit more about how it actually works, because it's quite important for the rest of the talk. So there are two parts to the CRISPR complex. So in blue here you can see something called Cas9. So this is a protein, and it's a special form of protein that can cut DNA. So it has two active sites, and it will bind to each of the strands of the double helix and will cleave each of these strands separately. And this generates what's called a double-stranded break within the cell. And it doesn't just do it randomly. Instead, it will find a particular target within the cell based on two different sequence motifs, or two different orders of the base pairs of the DNA itself. The first one is a little short sequence called a PAM. We won't spend too much time talking about this, but this is the part of the DNA which the Cas9 will bind to specifically in the first part. It's a very short 3 base pair sequence, usually. But if you like, the action is really contained within this so-called guide RNA sequence. So this is, as I said, it's the cousin of DNA. It's called RNA. And it has the ability to find its DNA match and base pair specifically with its DNA target. So there are very rigid rules around the building blocks and how they interact with each other. A always binds with T and C always binds with G in the 4 building blocks that make up DNA. RNA can also participate in this process, and therefore, even with a short stretch of, say, 20 nucleotides along this, or building blocks, along this molecule, it can find its specific DNA target in the 3 billion base pairs that are within the cell. So an amazing property. So how does it work? Let me show you a little bit more. It's probably easier to see this if we look at a video rather than just me explaining it. So this is the CRISPR complex. It's that RNA protein complex. This is this short sequence that the blue protein will bind to. So now we're looking at the Cas9 binding to it, and it's sampling that sequence along the DNA, shown here in green, next to where it's bound to, and it's looking for a match with this red guide RNA sequence. And in this case, it's found a perfect match. So, as I said, this is 3 billion base pairs for it to sort through. Once it finds it, it makes a double-stranded break. Happened quite quickly then, didn't it? Actually happens quite quickly within a cell as well. So there's a lot of specificity in this system. It's like a search and destroy. It'll scan the whole genome and it'll find the correct sequence and make a cut at that site. And I should say the thing that enables us to work very quickly with this is that we can change this guide RNA sequence very readily. We can easily swap in new guides and therefore change the region in the genome that it will target. Okay. So why is a double-stranded break important? Well, double-stranded break is important because cells really don't like having double-stranded breaks. You've broken the integrity of the very material. It's like ripping up a page from the book of life. They want to stick it back together because the cell won't be able to exist without having an intact book of life. So what the cell does is it uses its own repair machinery to repair that break, to stitch it back together. And this process is quite error-prone. In other words, what it sometimes does is it puts some extra building blocks in at the point of the junction, or even more commonly, it'll actually delete some of the bases at that site. So you've got this error-prone repair mechanism, which can, of course, change the sequence of a gene because you're either adding or deleting building blocks from the sentence. So that's really useful if you want to use CRISPR for therapy, because it means that we can target a disease gene and then inactivate it by changing the sequence within that gene using CRISPR. And we're going to show you two examples of how we can either inactivate a disease gene, in this case is a disease gene that causes blindness, or secondary, in a second example, I'll show you how we can actually fix a gene by cutting it again and then restoring the gene back to a much more normal state instead of a disease state. In other words, turning it from a nonsense into something that makes sense again. Okay, so what I'm going to tell you about on the next few slides are examples of so-called somatic gene therapy. And I'm going to talk about germline gene therapy later on, but this is not controversial on the left. On the right, very controversial. So I want you to make sure that we're in quite separate territories here. So we're going to start with the non-controversial, really useful, therapeutic, beneficial stuff. Now, the reason we want to go down this path is that by restoring a disease at the DNA level, it means that we're actually getting to the core of the problem. It means that potentially we could give someone a single therapeutic dose. If that worked really well, that would restore the gene back to a normal function again, and therefore that would give a lot of therapeutic benefit. That's why we're so interested in CRISPR therapy. It's not just treating the symptoms of the disease, it's treating the cause of the disease. Okay, so how does this work? Well, basically, we're looking to deliver CRISPR to the cells in the individual that have the problem. That might be a liver problem, it might be an eye problem, it might be a muscle problem. We're going to target those organs with versions of CRISPR and restore the gene back to a normal function. And these kinds of changes are not inherited by the next generation. They're just going to stay within the individual who's got the treatment. The first example I'm going to tell you about is one that we've been working on, and that's to fix a gene that causes an eye problem. This is called retinitis pigmentosa. Probably some of you in the audience have heard of retinitis pigmentosa. It affects around 2 million people worldwide, so not an insignificant problem. And essentially what happens in these individuals is that the rod cells that sense the light in the retina, in the eye, start to degenerate over time. Eventually they get to the point where they don't function anymore, they degenerate, and they cause the cone cells, their partners within the eye that also sense light, to also lose their function. So eventually, unfortunately, these individuals who are affected go blind, and there's not much that we can do about it at the moment. So we chose this as our disease target because we envisaged a way in which CRISPR could be applied to this particular group of disorders. And I'm going to show you just sort of one example of that. So sometimes this kind of disorder is caused by a gene that doesn't work very well. And we've got 2 copies, as I said, of every gene. There's one from mum and one from dad. So this is the version of the gene that doesn't work very well, and it produces a protein which not only doesn't work, but worse than that, it actually prevents the good copy from fulfilling its own function. So this is so-called dominant negative and a toxic gain of function. So those terms are just used to describe what's going on here, and this is a very attractive target for CRISPR, because if we can come in with CRISPR and specifically get rid of this mutant gene, that means that what we are left with is just the good version of the gene. And that means that the eye has enough rhodopsin, in this case the rhodopsin gene, has enough rhodopsin to carry the day, and it will function normally. That's the theory. So could it work? We wanted to find that out. So this is the way experiments are commonly done in the laboratory. So just to give you a bit of a sense of how we sort of approach these problems. This is the actual sequence of the disease gene here. So this is rhodopsin. And this is just that one building block, that one base pair that causes this entire problem. So it should read CCC across here, but instead it reads CAC, and this is the problem. So we knew that we could potentially target this area using CRISPR, and instead of just choosing one option, we decided to choose all of the options that we possibly could to get at this problem. So we wanted to test every possibility. So what I mean by that is that this guide RNA that I talked about earlier, that's part of the CRISPR complex, we changed this sequence to match either this one or this one or this one or this one. So we used everything we could throw at it to try and get rid of this gene. And because all of these changes span this region here, which is causing the problem, we hoped that it wouldn't affect the normal copy of the gene and would only hit the disease gene. So did it work? Well, the answer to that is when we tested this just in cells in the laboratory, so this is not in an animal, this is just in cells that we culture in the laboratory. The answer to that is it worked pretty darn well. So this is the kind of data we spend a long time looking at. So what we've done here is we've treated these cells with the 4 different test guide RNAs, as you can see on here. We've actually delivered the CRISPR machinery into the cell. The cells that didn't have any treatment at all are shown here on the right-hand side. So this is like the control, the starting point. And what you can see here is shown in peak is the amount of the disease gene within the pool of cells to start with. And this is what we're hoping to see afterwards. We're hoping to reduce this pink signal down to as small as possible, which means that we've gotten rid of the disease gene. And that's exactly what we saw. They all work slightly differently, but the point here is that we were really able to attack that disease gene very well. Which left the good copy of the gene to carry the day. So that looked really promising. Of course, we can't go straight from cells to working within humans as a therapy. We need to sort of build up further evidence that this could work in vivo, in the body, in the eye. So to do that, we have to use animals or preclinical models for these kinds of experiments. Now, just to make it very clear, anything we do in an animal experiment has to be thoroughly vetted and approved by our local ethics committee. We need to account for every animal that we use, and we need to do it in a way that's the most ethical way that we can do these experiments with the smallest number of animals as possible. Nevertheless, these kinds of experiments are necessary if we want to progress this into a human trial situation. So what we did in this case was that we took the CRISPR machinery and we wrapped it up in a virus. And this virus is called AAV, or adeno-associated virus, and it's commonly used for gene therapies in humans. We took the CRISPR machinery, wrapped it up in the virus, and then injected it directly into the eye itself. So we delivered it right to the organ that we wanted it to go to. And these mice were a very special kind of mice that have been engineered in a way that they express the human disease gene. So they have essentially the same kinds of phenotype, the same kinds of eye disease that we see in humans. So the question was, if we delivered CRISPR into the eyes of these, could we see if we got editing? Can we see if the CRISPR is working, and does it help to improve the vision of these animals? And that's what I'm going to show you on this slide. So this is really it's a very short, very long story made short, because these experiments are very, they involve a lot of work, a lot of careful analysis. But to give you a snapshot of how it worked, what you're looking at here is a section, so a very thin section, so a little snapshot of the retina tissue itself, so at the back of the eye. What you can see here in green, these green blobs, is actually the Cas9 expressed in the cells. So these are the cells within the retina that have the problem shown here in green, so that the CRISPR machinery is in the cell, which is great. And then we looked in the eyes to see if there was evidence of cutting. What you can see on the right-hand side here is that all of those little random deletions that I talked about earlier are being found within the cells within the eye of these affected individuals, and not within the untreated individuals. So we know it's the CRISPR going to work and doing what we hoped it would do. So that was a really exciting result to see. But what was really more exciting was that when we looked at the eye sensitivity to light in these treated animals versus ones that weren't treated, so untreated in red, treated in blue, you can see that we see an improvement in the vision in these animals. So it's very early days for our sort of journey towards CRISPR therapy, but we do have proof of concept for this particular strategy, and we're trying many others for these kinds of diseases as well. The second disease I'm going to tell you about, also work done in my lab, is a disease called Duchenne muscular dystrophy. Now, this is a more common disease, and I'm guessing, who's heard of Duchenne muscular dystrophy? Probably many. Yeah, it's one of the most common so-called single gene disorders that we know of. It affects boys because it's on the X chromosome, and there's no protective effect of a second X chromosome because boys are XY, and it affects around 1 in 3 to 4,000 male births. So really relatively common. And what's it's a very difficult disease, I think, to live with. And we've had some interaction with some of the patient support groups and so on for this disease, and it's really quite a devastating one to think about, because we start with probably a fairly normal individual in terms of starting to walk, but then there are mobility delays that occur early on. Usually loss of ambulation occurs around before sort of 12 years, 13 years of age, and a wheelchair is required, and then eventually due to muscle failure, these kids normally eventually die around 30 to 40 years of age. So there have been some advances in steroid therapies that have helped, but there's certainly no cure for this really devastating condition. So we wanted to help and think about ways that we could use CRISPR, and this is one case where we can use CRISPR, again using that cut strategy, but to use it in a different way to restore the function of this gene. So this one takes a little bit of mind-bending to work out, but hopefully you'll see how we're approaching this. So the dystrophin protein, the actual the fault here at the level of the protein is that dystrophin is actually like a shock absorber in your muscles. So your muscles are contracting all of the time, obviously, and it helps to protect the muscle cells from too much structural damage because of that contraction process. So it acts like a shock absorber. And if you don't have it, then the muscles get damaged over time. So it's a huge, huge protein, and it's encoded. The instructions for it is a huge, huge gene. It's one of the largest genes known in the genome. It goes on for like over 2 million of those building blocks in a row, so huge. And what often happens with this particular gene is that we lose whole words out of the sentence. So they're called exons, but you can think of them as words within the sentence, and we can lose them entirely. And that's a problem, because the way the gene is read is that it needs to fit together like a puzzle. So you can see they've either got sort of the pointy end pointing one way or the other, or they've got a straight flush end. And it's important that all the puzzles fit together in this way, a bit like a jigsaw puzzle. Otherwise the gene just doesn't make sense. So if we take out this exon here, so take out exon or word 44, that's a problem because 43 and 45 don't fit together. So that means that the gene's not going to make sense. It's not going to work properly. So here we turn to CRISPR. So again, using our trusty CRISPR strategy, and by just swapping in different sequences across this guide RNA, which matches the DNA, we could come up with strategies that would essentially target exon 45, the next exon. And we know if we cut twice with CRISPR, we can just get rid of that intervening sequence of DNA, which means that we can make exon 45 disappear, which means that now 43 connects to 46, and this is a good fit. Suddenly the protein makes sense again. It's a little bit shorter in the middle, but it makes much more sense to the cell, which is the important thing. So that's the idea behind this strategy. If you didn't get all of that, I'll try and explain it in a sort of grammatical way. So if we've got our normal gene, which is how to make a muscle really, really strong, then if we take out this R, the first R in really, it's how to make a muscle, and then it's, as I said, it's going to garbled after that. This is this frame shift that I referred to earlier. So now we come in with CRISPR, we get rid of this region, and the letters then sort of will read how to make a muscle really strong, which is great because the cell can interpret that again. The letters kind of shuffle along, it makes sense to the cell. And this is almost as good as the normal version of the gene. Not quite as good, but it's almost as good, which is enough to help that cell recover and prevent further degeneration. That's the idea. So it's a nice idea. Can it actually work? Well, so far, so good. So what we've done already is we've taken muscle cells from patients and we can grow them in the laboratory. And what we can do is then treat those muscle cells with CRISPR and ask the question, can we see dystrophin in those cells? Will dystrophin reappear in those cells because we've told the cell how to make dystrophin again by deleting this extra exon. So this again summarises a huge amount of work and a lot of experiments, but here's the control. So this is a normal individual. This is this huge protein. You're looking at bands on a gel. So what we do in the lab is we take the proteins and we run them through a gel which basically separates all of the proteins within the cell, and then we have a way of detecting just this protein specifically using an antibody. So this is what the normal control looks like. This is an individual who's got Duchenne's and hasn't been treated, and you can see there's no dystrophin, as you would expect. And here's a whole bunch of different strategies that we're using to try and restore dystrophin within those cells. And you can see sometimes it works better than others. You can hear it's worked really well. In these individuals, and these strategies, it hasn't worked quite so well, but the point here is that we can see dystrophin appearing again within the cell. So this is good news. Again, this is only work in cells. What we need to do next is to get evidence that it works in an animal. And again, with the caveats that I spoke to you about earlier about conducting experiments in animal models, this is the sort of latest results of this particular project, and it's looking quite promising once again. So here's a healthy muscle section. So dystrophin, as I said, is a shock absorber, and these are the cells of the muscle, and these are individual cells, or actually groups of cells that are fused together. And the shock absorber lives on the membrane, just on the inside of the cell. And it looks a little bit like a honeycomb pattern because that's where the protein dystrophin lives in the cell and does its work. So that looks great. That's the control. Here's an individual mouse that has a version of Duchenne muscular dystrophy, and you can see that the dystrophin is gone. And here's one that we've treated with, again, using this virus that delivers the CRISPR reagents to the cells in the animal. And we can see again, partial rescue, not complete rescue, but partial rescue of the dystrophin within those cells. And again, encouragingly, we can see that the muscle strength in these treated mice is better than the untreated controls. So more encouraging data that we want to take forward to the next stage. So in terms of what is the next stage, well, this is really, it's a very long haul to develop a drug indeed. And it's something I've never done throughout my career, but I'm very motivated to do it for these examples. But it really is quite a journey. So this is the point we're at the moment. So we've done the discovery phase, we've done preclinical research. If we wanted to take this into people, we need to do a lot of applications, as you would imagine, to get approval to do that. We're starting that process at the moment, but what's, I think, really important but daunting in some ways is that this is going to take several years to get this process going. It'll also take a huge amount of money, millions of dollars actually, to get the funding to do this. So if anyone in the audience has a couple of million to spare, I'd like to speak to you afterwards. If you want to come down, that'd be great. But yeah, it really is a very expensive, time-consuming process. And many drugs fail, of course, along this pipeline because they simply don't work once you get them into humans. Nevertheless, we want to give it a go. So just to really tell you about why, the bigger picture here, it's not just, of course, us working in our own labs on these kinds of projects. There's a whole lot of different CRISPR therapies that are being developed through different stages of the clinical trial process. But the poster child for this really is this new therapy, which, as I said right at the start, came out about 12 months ago. So this is an actual CRISPR therapy for a really devastating disease called sickle cell anemia. And there's not really time to go into the details of this, but just to tell you briefly how this works. Again, sickle cell is quite a common single gene disorder. It's quite a nasty disease because what happens is that the hemoglobin in the red blood cells has a predisposition to change its conformation, and it turns the cells from being a nice so-called biconcave disc that you would have learned about in Biology 101 if you'd done it, which carries oxygen, and they change their shape into this sickle cell shape. And it's really obvious if you take a blood cell smear from these individuals. They look quite odd. And what happens is that these cells tend to collect in the capillaries, and it's a very painful disease involving a lot of inflammation and shorter lifespan. So it's caused by a change in the dystrophin gene, in the hemoglobin gene, which encodes the blood-carrying protein. And it's a very clever little trick that the developers of this therapy have used. And basically, to cut a long story short, what happens after birth is that the adult hemoglobin that you and I have coursing through our veins is switched on. But until birth, you get by on what's called fetal hemoglobin, which actually is even a bit better than adult hemoglobin. And that's because it needs to get the maternal oxygen from the maternal system and transfer it to the baby. So it has even a higher affinity for oxygen than adult hemoglobin. And this is controlled by a protein called BCL11A. It's not important, but this is like a switch protein that controls this flip, this change of gene expression, this change in hemoglobin. And what happens with this CRISPR therapy is that they take the red blood cell stem cells out, they modify them so that they can no longer have this switch anymore, and this restores the fetal hemoglobin expression, and that works just fine in these individuals. So it really is a clever way of using what we know about genetics and gene regulation to restore the function of a gene. So the first person, one of the first people to be treated was Victoria Gray, and she described this as having, the life that I once felt I was only existing in, I'm now thriving in, and I feel like I've got a second chance. So this is, as I said, sort of our motivation as well. We want to give those with eye disease and muscle disease a second chance as well. So I'm pretty positive about somatic cell gene therapy, and there's a lot going on, as I said, and I don't have time to tell you about some even more recent developments on the technology side, but I just want to allude to it here, and just in one slide, just refer to a very new form of CRISPR, which is called prime editing. So instead of just being a pair of molecular scissors, prime editing goes one step further. It's not just a search and destroy, but it's like a search and replace. You can actually go into a cell and make very specific changes at the target site using prime editing. And this is really a game changer for not only genetics research, but also for therapeutics, because we can now think about targeting a whole range of new diseases using this kind of approach. So I'm really, encouraged by that. All right. So for the rest of the talk, I don't think we'll be able to have time to talk about invasive pests, but I'll be happy to talk about them afterwards if you like. But I do want to spend a little bit of time talking about the more controversial area of germline gene therapy. So the idea here is that we don't target an individual's organs that have the disease with the CRISPR, but rather we look at the embryo itself as the target, and before it starts to develop, we change the gene that would cause the disease in the individual if they were allowed to develop. So really looking at changing DNA at the very earliest stages of life. So it's quite controversial, as I said. So can we even edit human embryos? Of course, this is a tightly regulated area. And what are the risks associated with this kind of approach? So I just want to tell you a short story about, I guess, our involvement in this area. We don't do any work on human embryos. In fact, in Australia, it's illegal to do any work on human embryos at all. And instead, we have been involved using mouse embryos to look at something which was reported in human embryos a few years ago. So this is the story. It came out in Nature back in 2017. And essentially, they were looking to do proof of concept for human germline editing. And what they did is they made embryos using IVF that had a disease gene and a normal gene. And they used CRISPR to target the disease gene. And what they reported was that, in fact, surprisingly, the normal version of the gene had been used to repair the double-stranded break to create two normal versions of the gene. Now, this was very surprising to many, including us, and we thought perhaps they might have missed something. And in fact, what they might have done is to just create a big hole in the disease gene. And because they only looked at this particular region of the gene to see whether the CRISPR had worked, it might be that they were missing this large hole because they were essentially not looking for it. So we repeated their experiments in mouse embryos, and we were able to show that, in fact, a large proportion of the time, almost 50% of the time, we see that, in fact, when you use CRISPR, in that context, it'll make a large hole in the chromosome. These span many, sometimes thousands of bases, and it's since been shown that they can span even millions of bases, even taking away half the chromosome. So this is just really highlighting that safety remains a sort of major aspect of this kind of approach. Before we even think about using it, we need to know 100% what the consequences are of the CRISPR application within this, within human embryos. So there are many issues associated with this kind of that I think you would be aware of. Why would we even want to use CRISPR? So one argument against using CRISPR, which I think is quite persuasive, is that we already have a way of determining whether an embryo contains a genetic mutation or not, if we know in the family that a particular genetic change that causes disease is present. And the way that's done currently is to do so-called pre-implantation diagnosis, where we take just one cell of an embryo before they are potentially transferred to the female. And we look at the genotype. So we look at the DNA and ask, does it contain this known mutation or not? And if it doesn't, then we think it's safe to put that embryo back. So this is a really powerful technology. The embryo doesn't hurt the embryo, it recovers from having one cell removed. And it's used quite commonly, routinely in clinics already. So that's one reason. So instead of trying to change the DNA back to something normal, we can already just detect the embryos that don't carry it and use those instead. There are ethical issues associated with germline changes. For example, informed consent. So how can an unborn embryo give consent for the procedure when it obviously that's not possible? So these are the things that ethicists dwell on, and I think it's a very valid thing to be thinking about. We can never eradicate genetic disease. That's another argument that's often used to put forward about using CRISPR in this context. We can't eradicate genetic disease because it'll always be happening in other families. We can't stop that process. It happens all the time. Mistakes are made. And even if we cure a genetic disease in one family, it will emerge in other individuals within the population. That's just the way it is. So we can never completely eradicate genetic disease. Designer babies is one that comes up often. So could we enhance intelligence? Could we increase in sporting ability, change appearance? All of those kinds of traits, all of those kinds of features are hugely complicated. They're not the kinds of things that we could just change with one gene and hope to improve someone's intelligence. So I think it's really, we wouldn't actually even know where to start with that kind of approach. So I don't see that as something that's on the horizon even remotely. As I've already talked about, safety and unintended mutations, and also the fact that in Australia, any of this kind of research is currently illegal. So you wouldn't imagine that anyone could actually CRISPR an embryo, a human embryo, do you? I mean, has that actually happened? Well, the answer to that, surprisingly, is yes. So this was a story that came out a few years ago. It's not published, but this is data presented at a conference. This surprised all of the CRISPR community. In fact, I think anyone who has come across this story. Can I ask quickly, who's heard of this story already? Yeah, about half the audience. Okay. So what happened was that at a conference, a CRISPR conference, Dr. Hay here in China had reported that he had used CRISPR to delete the AIDS receptor in 3 individuals. As I said, it's not published, but at the conference he reported data that looked very convincing. And he told the audience that he had manipulated the embryos and these individuals were born. And his justification was the father had AIDS and he was looking to prevent the offspring from getting AIDS by introducing a common mutation that we know of that is relatively benign. But that's not really the point. The point here is that he has gone ahead and done an experiment that is illegal under all codes in all countries. And it turned out that he had not received proper ethics approval, of course, for this experiment. The editing, in fact, it emerged wasn't perfect. We don't know much about the children that were born. And the consequence ultimately was that he went to prison for 3 years. So I think this is a very important lesson to learn, that it really did horrify the CRISPR community and only reinforced that we need to be very, very careful and cautious before going down anywhere close to this kind of line. So with great, CRISPR is very powerful, but it also brings great responsibility as well. So I think I'll just about wrap up there because we're out of time. But what I will say is that I'm really, really enthusiastic about CRISPR somatic gene therapy. I think it's got a huge amount of potential. And as you've seen, we're well on the way in many regards. And there's already a therapy that's out, which is great news, I think, for those with sickle cell anemia, although it is very expensive. I'm happy to talk about that as well. And that there's not time, unfortunately, to go through this. But as I said, I'm happy to take questions. But the last thing I want to show you is that CRISPR, I've really just scratched the surface. CRISPR is being used in all sorts of applications beyond what I've been able to tell you about, including in the agricultural world. So not just for drug development, for agriculture, biotech, drug discovery, biofuels, all sorts of applications. Anything that's got DNA, if you can change it with CRISPR, then you can start to develop new ways of doing things, and that has a huge amount of product potential. So with that, I'd like to close and really thank the most important people, the people that actually do the work, the people that are sitting at their desks doing bioinformatics or in the lab doing stuff with CRISPR in cells and with animals as well, because that's really where the action is. So I've got a really great team that worked on all of the projects that I've talked to you about today. And also I would like to thank funding from these organisations as well. We can't do any work without funding as well, and that's really important. So with that, I'll thank you for your attention and happy to take questions.
Hello. Sorry. Thank you, Paul. We have a couple of minutes for maybe one quick question. So we can open it up to the audience and to online. But did anyone in the audience have a question?
It's perhaps a foolish question, but if you're wanting to fix a retina or change something, you have to apply CRISPR to every cell in the retina. How are you sure that you're doing it to every cell and not just a few? Yep.
It's a really good and important question. So there's sort of 3 things that have to happen if you want a CRISPR therapy to work. You have to deliver it to the right cells. It has to make a change in those cells that's beneficial, and you don't want it to be making changes in other parts of the genome where it could have a negative impact. So one of the most important things is the delivery side, and delivery efficiency is something that everyone's really focused on maximising. So in the case of the retina, we can access it relatively readily by injecting straight into the eye. Now that sounds kind of terrible, but eye injections are commonly used for diabetes treatment, for example, for neovascularization. So that's actually vitreal injection is relatively common, and we can actually inject it subretinally, so under the retina as well. So we can get it to the right place, and then it's kind of up to the virus to find as many cells as possible. And that's where there's also research in changing those viruses subtly so that they've got to maximise their chance of getting into the right cells. And that you can do that by changing what's on the outside of them. So that's a reasonably well-trodden path. But now there's also emerging a whole different range of delivery tools, for example, so-called little nanoparticles. So these you can make in the laboratory, package them within your CRISPR. So, for example, Moderna vaccine, for example, we would have had a lot of Moderna vaccines, and the Pfizer vaccine, they are mRNA vaccines. So they're basically bits of mRNA wrapped up in this particle that we inject into muscle. You can use exactly the same kind of particles to target the retina, or indeed any other cell that you like as well. So the short answer to your question is there's lots of technology going on, development, to maximise the delivery so that you can get the most bang for your buck, which is essentially where you were going. Yep.
I think we have time for one more quick question. If anyone's got one.
Just talking about delivery, what about delivery across the blood-brain barrier?
Yes, I didn't have a chance to talk about this. We work on an epilepsy project as well in the group, and there is a CRISPR therapy opportunity for that project as well. And there are some versions of the viruses that I talked to you about that are pretty good at getting across the blood-brain barrier. They're not perfect, but they're pretty good. The group spent a lot of time optimizing and trying to get the maximum amount of the CRISPR wrapped up in the virus across that blood-brain barrier, because for those of you unaware, there's like a barrier around the brain that prevents drug delivery a lot of the time. It's a very sort of selective barrier that most molecules can't get through. So you need to get around that problem, and it is possible using these viruses. So, yep.
We'll wrap up question time now, and I'd like to finish by thanking tonight's wonderful speaker, Professor Paul Thomas, and thank you to you, our audience, for viewing and for your questions and comments. Be sure to sign up for our mailing list to receive the latest information about Research Tuesdays, and I thank you for tuning in once again, and we hope to see you next time. Good night.